We know from family, twin and adoption studies that physical and behavioural characteristics have a substantial genetic component. Despite this, we are still far from being able to make accurate predictions about people based on their DNA alone. The gap between the heritability of individual differences which we can measure and our ability to account for this genetically is because of a number of statistical and technical challenges in modelling the influence of DNA on complex human traits. This video is largely based on the paper by Nadav Brandes and his colleagues at the Hebrew University of Jerusalem. If you are comfortable reading scientific papers, I encourage you to check it out as it is a great read and completely open access. Link to the paper is in the description. But otherwise, I'm going to try and explain its key points in this video in a way which I hope everyone will be able to understand and take away some perspective of the sheer complexity of this field. Perhaps the most fundamental issue in human trait genetics is how we go about quantifying the specific characteristics or traits which are to be investigated. In a biological context, this is called phenotyping. Anything about an individual which can be measured, such as their height, body weight, eye colour, skin colour, personality, intelligence, etc., can be a phenotype. Quantitative phenotypes are those which can be measured on a continuous scale, such as height, whereas categorical phenotypes are characteristics which a person either has or does not have, such as a certain eye colour. Some phenotypes are relatively straightforward to measure. Height is the distance between the top of a person's head and the soles of their feet. Eye colour can be observed visually, but many characteristics cannot be directly observed. For example, it is a lot less apparent how to go about quantifying more abstract traits about a person, such as intelligence or personality. Great efforts have been made into constructing measures of abstract phenotypes which are valid and reliable. Validity refers to the extent which a test of a trait measures what it purports to measure, and reliability refers to the extent which a test will give the same results for the same individuals when applied on different days or by different practitioners. Valid and reliable tests to measure abstract traits do exist, but they are not as straightforward to interpret as measures of physical characteristics. Personality tests, for example, are valid in the sense that they can predict patterns of behaviour across a person's lifetime, but they are not able to accurately predict what an individual would do in any given situation. In terms of reliability, although personality tests tend to give similar results for the same person when taken on different occasions, the exact results are expected to vary within a certain range. Just as there are complications in measuring quantitative traits, similar issues exist when it comes to classifying categorical traits. Certain diseases are commonly thought of as purely categorical, whereas they could be more accurately characterised as existing on a continuum of severity which is not always straightforward to quantify. For example, we think of people as either being diabetic or free from diabetes, but there actually exists a continuum of glucose sensitivity on which, after a certain threshold, a person is considered to be diabetic. In physiological conditions, although thresholds like this are arbitrary to an extent, they are convenient in the sense that they can be clearly defined and measured in objective tests, such as the fasting blood glucose test, allowing standardisation of diagnoses across practitioners. Diagnosing non-quantifiable phenotypes, such as psychiatric disorders, cannot rely on well-defined thresholds. That is not to say that psychiatric diagnoses are unobjective. Reproducibility of diagnoses between practitioners in psychiatry is in fact similar to that of other medical fields, but rather that it is a more complex process and requires an element of subjectivity. Aside from measuring or classifying phenotypes, another consideration is how phenotypes are defined in the first place. There are many cases in which two or more phenotypes are treated as separate phenotypes when they are actually manifestations of the same underlying biology. Take schizophrenia and bipolar disorder, for example. These are treated as two distinct yet related psychiatric diagnoses. However, they have strongly overlapping genetic determinants, leading some to argue that they should be seen as residing on a single psychosis continuum. Conversely, some phenotypes could be composed of many subphenotypes which either manifest in a way which make them appear outwardly similar to each other, or in a way which make them pragmatic to be grouped together for the purpose of treatment. In these cases, all subtypes are classified together as one phenotype. However, their underlying biology may be completely unrelated to each other. For example, there are subtypes of depression which are grouped under the same umbrella term because they manifest in symptoms which are outwardly similar, despite having distinct biological mechanisms and requiring treatment specific to each subtype. Autism spectrum disorder covers such a wide range of behaviours that two people who have received a diagnosis of autism may have little in common in terms of their presentations. However, the diagnosis is convenient for the purpose of ensuring that they have access to the additional support that they need. Imperfect validity and reliability in measuring abstract phenotypes, subjectivity in classifying non-quantifiable phenotypes, and a degree of arbitrariness from a biological perspective when it comes to phenotype definition are all potential windows for statistical noise and bias to sneak in when searching for correlations between phenotypes and genetic variation. The presence of statistical noise is expected to reduce our ability to detect associations between genetic variants and phenotypes. However, that does not mean that we should throw the baby out with the bathwater by giving up on difficult to measure phenotypes. Genetic studies are already giving us meaningful leads in understanding the biological underpinnings of phenotypes such as the examples given previously. And besides, no phenotype is ever perfectly measurable to begin with. Even phenotypes as seemingly solid as height vary within the same individual over the course of a day. That does not mean that the data are not meaningful. Increasing availability of datasets with transparent information about how phenotyping was carried out can provide opportunities for researchers to fine-tune the definition of the phenotype they are using, and thereby reduce statistical noise and improve the clarity of associations between phenotypes and genetic variation. That being said, even if hypothetically perfect phenotyping data were available, this would only solve half the problem. Pinning down genetic variation is a challenge in itself. A genome is the complete set of DNA which a person carries in the nucleus of each of their cells. It is comprised of strands of DNA divided into chromosomes. A single unit of DNA is called a nucleotide, and they come in one of 4 types: guanine, thymine, cytosine, or adenine, or GCTA for short. Each person has more than 3 billion nucleotides in their genome, and the vast majority of them are in the same order from person to person. However, a small percentage of these nucleotides are expected to vary between individuals. These are called single nucleotide polymorphisms, or SNPs. The process of finding out which SNPs a person carries that is used in most studies is called genotyping. This is different from genome sequencing, which reads almost every individual nucleotide in the person's genome. Genotyping instead reads only a select few nucleotides and uses this information to statistically impute which SNPs a person carries. How this works is beyond the scope of this video, however, check out one of my previous videos for more information about imputation. Genotyping is typically used because it is way cheaper than whole genome sequencing. Despite the economic advantages of genotyping, this introduces some technical issues. For example, rare SNPs are typically not accurately imputed from genotyping data. The reason for this is because imputation relies on reference panels of genomes from individuals who have undergone whole genome sequencing. Rare variants, by definition, occur infrequently in the population and therefore are underrepresented in reference panels. Typically, an arbitrary threshold of 1% population frequency is set for SNPs to be included in studies to ensure that imputation has been carried out to an acceptable degree of accuracy. The absence of variants which occur at a lower frequency than this threshold is expected to have a substantial impact on the potential to capture the influence of genetics on phenotypes because, due to natural selection, variants which have a strong impact on phenotypes are expected to be rare. Over the years, whole genome sequencing has been getting cheaper, although the decline in cost has started to plateau in recent years. Despite this, new whole genome sequencing datasets have become available recently, such as the UK Biobank whole genome sequencing cohort of 500,000 individuals. Increasing availability of data such as these has the potential to unlock some of the missing heritability by bypassing the need for imputation. However, this won't necessarily solve the rare variant issue. Studies require very large sample sizes to identify associations between phenotypes and rare variants due to the low frequency of rare variants. The issue of the lack of statistical power to detect associations with rare variants could potentially be tackled by grouping variants together as a single variable based on the gene which they affect. However, as will be discussed later in the video, finding which gene is affected by a genetic variant is not so straightforward. Once you have a large sample of individuals who have been phenotyped and genotyped, you might expect that finding genetic variants which are involved in the development of a phenotype should be a straightforward matter of running the stats. However, genetic data are more complicated than most other types of data because of biological factors which need to be considered. The main method of identifying associations between phenotypes and genetic variants is called a genome-wide association study, or GWAS for short. If you are unfamiliar with genome-wide association studies, please check out my video series about them on this channel, but simply put, they are tests of correlation between phenotypes and variation in DNA across the whole genome. A GWAS about height, for example, would look at the DNA of thousands of people and measure the frequency which genetic variants occur in people based on their height. A genetic variant which is common amongst taller people but rarer in shorter people would be considered to be associated with height. That being said, the phrase correlation does not equal causation needs no rehearsing. There are several complications which need to be dealt with to sift out genetic variants which actually cause the phenotype from the list of associations you find in a GWAS. During recombination, the process in which genetic information is swapped between the sperm and the egg during reproduction, genetic variants which neighbour each other in certain locations of the genome are more likely to stick together than genetic variants in different locations. This is a phenomenon called linkage disequilibrium, and it works in a similar way to how, in a roughly shuffled deck of cards, the cards which were next to each other before shuffling are more likely to wind up next to each other after shuffling than to any of the other cards in the deck. This has implications for identifying true associations from the results you find in a GWAS. The results of a GWAS can be visualised in plots like these, which are called Manhattan plots. Each of the dots across the x-axis represents a genetic variant across the genome. The y-axis represents the negative log of the p-values. Basically, the higher up on the y-axis the dot is, the more confident we can be that the genetic variant is associated with the phenotype. You will notice that around each peak on the y-axis there is a cluster of neighbouring dots surrounding it which are also high on the y-axis. This is a result of linkage disequilibrium. So for each cluster you see in the results of a GWAS, it is likely that only one of the genetic variants are truly the variant which causes an increased likelihood of the phenotype, and the rest of them are just along for the ride. It is tempting to assume that the lead SNP, the most significant SNP out of a cluster, is the causal SNP. However, this is not always the case. Finding the true causal variant among them is often a complex problem. The process of identifying the causal variant or variants out of a cluster of statistically significant variants is called fine mapping. Fine mapping works by applying Bayesian statistical methods to estimate the probability that each variant in a cluster is causal, and by annotating variants using databases of known functional sites of the genome. Comparing GWAS results from multiple ancestries is another method of fine mapping, as patterns of linkage disequilibrium tend to vary more between individuals of different ancestries. Therefore, variants which maintain high significance across multiple ancestries are more likely to be causal. Despite the great progress which has been made in developing methods for fine mapping, it is not always possible to determine which variants are causal with certainty. Once a causal variant is identified, it is a whole other challenge to determine the actual mechanism by which the variant influences the phenotype. Even among known causal variants, few have a known biological mechanism. Due to the nature of complex phenotypes, causal variants are most often not found in the coding regions of the genes, the sections of DNA which are transcribed into proteins, but rather are usually found in the regions of the genome which regulate the expression of genes. The genes which are affected by a regulatory region are often nearby the regulatory region, but not necessarily. Sometimes the regulatory region is far from the gene, and there could be many genes which are closer to it than the gene which is actually regulated. Sometimes the gene is even on a different chromosome altogether. To complicate matters further, a genetic variant could be associated with more than one phenotype, meaning that it is not always possible to tell if a genetic variant affects the phenotype directly via biological mechanisms, or indirectly affects the phenotype by the influence it has on another phenotype. To illustrate with a hypothetical example, if a genetic variant was found to be associated with both self-confidence and perceived attractiveness, it would not be possible to tell if the genetic variant independently causes both increased attractiveness and increased self-confidence, or if it impacts attractiveness and thus also transiently results in increased self-confidence. One method of discovering which genes are influenced by a specific genetic variant is to compare genetic variation with gene expression. Take a sample of a tissue from individuals with one version of the genetic variant, and a sample of the same type of tissue from individuals with the alternative version of the genetic variant, and compare the expression of genes in that cell using RNA sequencing. RNA is the form of nucleic acids which are transcribed from genes in cells. Genes which show a difference in the level of expression between the two groups would be prime candidates for genes which are regulated by the genetic variant. A key difficulty is that datasets containing RNA sequencing data and matching phenotype information are not always available for the tissues relevant to the phenotype, and it is not always possible to generate these data by conducting experiments on humans. This is especially true for behavioural phenotypes where the expression of relevant genes is expected to be in the nervous system, particularly the brain. For some phenotypes, it is not even obvious which tissues are relevant to begin with. That being said, while identifying causal variants is useful for understanding the biological mechanisms by which genetic variation impacts variation in phenotypes, it is not necessary to identify causal variants to be able to make predictions about people's phenotypes from their DNA. Polygenic risk scores, or PRS, are models of predicting phenotypes using the results of a GWAS, which are completely agnostic about which variants are causal. A PRS for a phenotype is calculated by summing the effect sizes of each associated variant. The effect size is simply put how much of the variance of the phenotype can theoretically be explained by the variant, multiplied by the number of copies of the variant a person has. A person can have 0, 1 or 2 copies of any genetic variant. Do this for thousands of individuals in a population and compare their scores with their actual phenotype, and you can assess the predictive power of your PRS. If you make a PRS for height, and most people who have a high polygenic risk score are taller than those who have a low polygenic risk score, then you can say that your score is valid. By mapping the distribution of the PRS in a population against their actual height, you can even start to make predictions about how tall someone is based on their DNA alone. How much predictive power your PRS will have will depend on several things, such as the sample size of the GWAS the effect sizes were calculated from, the extent to which the phenotype is genetically influenced in the first place, heritability, and as touched on previously, the validity of the definition of the phenotype. The problem with polygenic risk scores is that performance is poor in predicting the variation in a population for most phenotypes. There are many potential reasons why polygenic risk scores do not capture the bulk of the heritability expected from the results of twin and family studies. One of the suspects is the underlying assumption of additive relationships between genetic variants and phenotypes. When calculating a polygenic risk score, the effect sizes of the genetic variants are usually summed. However, in reality, we know that this is not how genetics work. It is entirely possible that the variants which affect a phenotype in a certain direction could have a completely different effect in the presence of another variant. For example, genetic variant A could have the effect of modestly increasing height in the presence of genetic variant B, but in the presence of variant C, in the place of variant B, it may have a larger effect, no effect, or even affect height in the opposite direction. Ignoring the possibility of such interactions inevitably limits the predictive power of polygenic risk scores, but it is necessary to do so. The sheer bulk of computational work required to assess the effects of interactions between genetic variants across the whole genome is simply not feasible. Imagine that we genotype 10 million SNPs in a sample for a GWAS. If we were to examine the interactions between every possible pairing of SNPs, this would yield 10 to the power of 12 statistical tests. That's over 50 trillion tests, and that's only assuming interactions between pairs of SNPs. The number of possible interactions when you include 3-way, 4-way, etc. interactions are astronomical. Even if we had some ungodly powerful computer to compute all of these tests, after correcting for multiple testing, the sample sizes required to detect these associations would outnumber the population of the Earth. Despite the limitations of assuming an additive model, there is some evidence to suggest that an additive model for complex traits may be true enough that its advantages when making predictive models may outweigh its costs. Meta-analyses of twin studies find that identical twins are on average twice as similar as non-identical twins for the majority of phenotypes. As identical twins also share twice as much DNA compared to non-identical twins, this provides support for the idea that the additive effect of genetic variation overwhelms the effects of interactions between genetic variants. Another piece of evidence in support of the idea that interactions between genetic variants is minimal is that attempts to use machine learning models which do not assume additivity have produced worse results when making polygenic risk scores, despite performing well in producing predictive models using non-genetic data. This may indicate that the effect of interactions between genetic variants is minimal. However, it may also simply be the case that off-the-shelf machine learning models are not best suited for genetics, and it may be necessary to develop less generic machine learning methods which are better suited for genetic data to properly incorporate the effect of interactions between genetic variants. Despite the lack of success in modelling interactions between genetic variants, The additive model should remain an immediate suspect for the shortcomings of genetic prediction models. Similarly, another potential source of missing predictive power is the disregard of potential interactions between genetic variants and environmental factors. The polygenic model of genetic influence on phenotypes assumes that there is a certain level of risk of developing a phenotype conferred by genetics and a certain level of risk conferred by environmental factors, and that the sum of these two categories make up a person's total likelihood of developing the phenotype. This assumption ignores the possibility that certain genetic variants may only influence a phenotype under certain environmental conditions, or that the presence of certain environmental factors may modulate the effect of genetic variants to have an effect in the opposite direction when the environmental conditions are absent. For example, genetic variants which cause less skin pigmentation may be found to be associated with likelihood of skin cancer if the study was carried out on individuals living in an environment with high sun exposure, but would be irrelevant to skin cancer amongst people living in environments with low sun exposure. It may be possible to improve the performance of polygenic risk scores by controlling for the presence or absence of environmental factors. For example, it would be foolish not to control for smoking status when carrying out an association study into lung cancer. But when relevant environmental confounders are not known, it is not practical to control for every possible combination of environmental factors. Despite the difficulty in understanding the interplay between genetic and environmental factors in the development of phenotypes, this subject is of special interest to many scientific fields, especially psychology, where gene-environment interactions are believed to play a central role in human personality and behaviour, and an active debate is still going around the old question of nature versus nurture. Another major issue is translating polygenic risk scores generated from GWAS data from one ancestry to individuals of another ancestry. Earlier, I compared the source of linkage disequilibrium to shuffling a deck of cards. This is a bit of an oversimplification, as patterns of linkage disequilibrium vary between different ancestry groups. So, as the genomes of individuals from another ancestry group have been shuffled in a different way, genetic variants which are associated with the true causal variants in the sample population from a GWAS may not be linked to the same causal variants in individuals of a different ancestry. This is a near fatal problem for the application of polygenic risk scores in a healthcare context, as since the bulk of genetic data so far has been collected on individuals of European ancestry, the best performing polygenic risk scores are not applicable to the majority of the population. One of the avenues towards solving this problem is to develop independent polygenic risk scores for each ancestry group. This is becoming increasingly feasible as the amount of non-European genotyping data continues to increase. However, this still does not account for the millions of individuals in the world who have complex ancestries. One method of tackling this problem is calculating local ancestry polygenic risk scores, that is, applying ancestry-specific PRS to the sections of the genome inherited from each ancestry in mixed ancestry individuals. This is showing some promise. An alternative is to develop polygenic risk scores using only variants which are presumed to be causal variants using fine mapping methods. However, this method risks sacrificing too much signal for the sake of eradicating noise. It is necessary to control for ancestry when identifying genetic associations in the first place. One reason for this is that correlations between genetic variants and certain phenotypes may reflect environmental causes of the phenotype associated with genetic variants which are common in a population which happen to live in the environment, rather than an actual causal link between the genetic variant and the phenotype itself. To give an absurd example, there are presumably no genetic variants which cause you to be more likely to use chopsticks. However, if you were to carry out a GWAS into chopstick use without taking the precaution of controlling for ancestry, you would find significant associations with genetic variants which are common in East Asian populations. GWAS are usually carried out in a single ancestry group at a time. However, it is also necessary to control for residual ancestry, genetic differences between subpopulations within ancestry groups, to prevent spurious results similar to the chopstick problem. For example, certain parts of the UK have higher prevalences of disease than others due to many factors such as differences in climate and sociological factors. A GWAS into heart disease amongst UK citizens, for example, might find significant associations with genetic variants which are common in the west of Scotland, but this may reflect the history of poverty on health in this region rather than actual causal effects of the variants themselves. This video provides a broad overview of the challenges in understanding the relationship between genetics and traits. The technical and theoretical problems are even more complex still in their finer details. Understanding genetic associations is never going to be straightforward, as genetic associations are not meant to represent permanent deterministic causes of phenotypes, but rather fluctuate across time, environments, and populations. Researchers in human trait genomics understand this and, rather than trying to pin down the genetic causes of phenotypes, attempt to gain a deeper understanding of the fluid relationship between DNA and human health, disease, and characteristics. Our ability to do so is inseparable from the technical problems associated with defining and measuring phenotypes, capturing genetic variation, identifying genetic association, sifting out causal variants, and accounting for ancestry confounding. Methods of dealing with these issues are improving all the time, but as in all scientific disciplines, success depends on clear communication between researchers and clinicians and a universal agreement on the issues that need to be prioritised.